So much for coming to listen to my talk. I'm Dr. Keely Hammond. I'm in the final year of the Medical Microbiology and ID training program at McGill. And on behalf of myself and also some of the other staff doctors I work with, including Dr. Cedric Yansuni, I'm going to talk about rapid phenotypic antimicrobial susceptibility testing and how it can hopefully change our clinical practice. So these are my disclosures. So I want to talk about a few things here. I'll talk about why we perform AST, what the conventional workflow looks like. We kind of touched on that yesterday in the afternoon discussion. I'm going to talk about already commercialized rapid AST technologies and also some non-commercial technologies that are just very exciting to talk about, and we'll get a little bit into the details of how they work. So as a human veterinarian, as we heard earlier, I'm most interested in understanding what's the best treatment for an individual patient for their pathogen. And there are other reasons why we perform AST, but the need for the rapid testing is really most relevant for patient-level decision-making. So we assume by doing the AST that we're predicting the treatment outcome with a given agent. So that's kind of why we're doing this test in the lab. So we discussed already how AMR has a big impact on Canadians, but it has an even bigger impact in other parts of the world. As we can see in sub-Saharan Africa, there's almost twice the rate of deaths attributable to AMR. And despite this, the laboratory access is extremely minimal. So very few countries have any access at all, and those that do, many don't have access to AST that could potentially guide them towards appropriate therapies for their patients. So I'm going to get back to why it's important to consider this context as well as the Canadian context when we talk about new AST technologies. So the conventional workflow looks a little bit like this. So we start with a patient sample. I'll take blood as an example. We put it in a blood culture bottle. It goes in the incubator. It becomes positive, and then we have to isolate a pure colony before we do any type of AST testing. And so this whole process can take up to 72 hours, as we discussed yesterday in the afternoon. And then there's a variety of conventional methods that we use. We saw that picture of the agar plate with the little discs on it, so that's disc diffusion. Gradient diffusion strips, or e-test, is another method. That's the strips on the little agar plate. And then there's a variety of other methods that are more automated but can still take time and still have to integrate into that lab workflow where technologists may not be working 24/7. So the real pros of the conventional workflow are the fact that it's super flexible. So you can put in almost any specimen type because you're going to start by isolating a pure colony and then you're going to put it into your AST. And it's also very well, we have a lot of experience with it. There's some good validated interpretive criteria, lots of different workflows, and it's phenotypic, which is super important. So phenotypic meaning we put the bug with the drug and we see what happens, as opposed to making assumptions based on resistance genes, for example, simply because we don't have a clear enough understanding of those genetic inputs in the bacteria yet to be absolutely sure that we're doing the right thing if we pick a certain bug-drug combo. But it is a slow thing, so it can take up to 72 hours, as I mentioned, and it requires that pure colony to be isolated first. And it really requires a pretty complicated lab and a pretty complicated supply chain. So as I mentioned already, with the phenotypic testing, this is sort of our gold standard of testing in the lab to make individual patient-level decisions, because we're not making any assumptions about genetics here. So the main limitations really are the slowness of the workflow and also the fact that it requires this complex infrastructure. So the lab requires really highly trained personnel as well as this proper quality control, proper reagents, all this that's very challenging to implement even in Canada, let alone in limited resource settings. So how can new technologies change this? So for sure, we can try to reduce the time to result, which should reduce the time to most appropriate therapy. And we can try to simplify the workflow by constructing tests that we could use in limited resource settings. And so there's a number of ways to reduce time to result. I'm going to sort of get into that with some specific examples later on. And then in terms of simplifying the workflow, the core elements there really are to test directly from a specimen. So instead of having to isolate pure colonies from your initial specimen, just to test directly from the blood, for example, or the urine, and to streamline all the analytic steps within the same instrument. So these are some strategies to reduce time to result, sort of detailed out from a review article that I collaborated on with Dr. Yansuni and Grace Rosetnik and the rest of our team. So these are a few sort of methods to reduce the time to result, and you can also see that there's many technologies that use more than one method. And I want to also look at prior successes in workflow simplification for diagnostics. So we already heard about how other infectious diseases have been more successfully managed than AMR on a global scale. So I'm showing you here some examples of rapid diagnostics tests for HIV, malaria, and TB that can be used in these limited resource settings. And I hope that's like a uplifting or inspiring kind of thought with respect to AMR, given that it's just trickier with AST because it's a lot more complicated as a test to simplify. But we know at a global scale, at least in the past, there have been successes with diagnostics with the right funding and government and commercial partner support. So I'm just wanting to reference this review article if you're interested in looking at this topic on a more detailed level. I'm going to kind of present here some highlights, but this is really a good overview of many different commercial and non-commercial tests that are available right now. So I'll start by talking now about, in more detail, some of the commercially available technologies. So I'm kind of grouping them into three groups. So direct from urine, direct from the positive blood culture, and then direct from positive blood culture with integrated species identification. And the integrated species identification is important because it's actually necessary for us to interpret that AST result to know the actual species of the bacteria. So why are these two types of tests important? So urine is important because it's a really high volume test, and it also has a really uncomplicated matrix, the matrix being the stuff that's not the bacteria. So it's pretty easy to handle. Blood, on the other hand, is a very important clinical specimen. The matrix is more complicated, and so you have to kind of deal with that matrix as well as try to get a fast result. Okay, so here's an example of one technology approved in Europe. So it takes urine specimens and uses kind of advanced imaging techniques when the urine is combined with antimicrobials to look at bacterial growth, and it has a super fast time to result. 45 minutes. And the actual result it gives is just a susceptible or resistant. It doesn't actually tell you the species, but it's equipped to handle 5 different bacterial species and 5 different antimicrobials. So we kind of do want reporting of a little more than just susceptible or resistant. So first of all, we do want to know the species because it helps us interpret the susceptibility result. And this test doesn't actually show us something called the MIC, or the minimal inhibitory concentration. So this is a number telling us at what concentration the bacteria is effectively inhibited. And if we don't know that number, it's actually quite hard to compare between tests or to reinterpret those numbers if we make some changes to what we think is the cutoff. So that's some drawbacks of this particular test, but it's exciting to see that it's such a fast test. So in the direct from positive blood culture space, there's actually quite a few options. So the more sort of straightforward option is something called disc diffusion with the positive blood culture used directly as the inoculum. And then there's also a whole variety of automated systems that use positive blood culture as their input. So here's a picture of that disc diffusion. So basically what we would normally do is we would first get the positive blood culture, we would put some of that broth on a plate, and then we would see some colonies growing, we would take a pure colony and then do this disc diffusion that you see here. But here we just take that blood culture broth, we put it directly on the plate, we incubate it for a certain amount of time, and we read the plate directly, which can really cut down up to 24 hours off of the whole workflow. So this is pretty straightforward, seemingly. And there's sort of 2 big sort of regulatory or advisory bodies for microbiology, UCAST, which are the Europeans, and CLSI, which is the US, and we often follow CLSI ourselves. So they have actual protocols for doing this and specific organisms where this test has been validated. And you can actually use this as a final result as long as you have the quality parameters are met for the test. Like, for example, you can't have 2 different bugs growing in your one bottle. Now, so the time to result is really, like I said, can be reduced by up to a day. But this is a test, while looking quite simple on the surface, requires that whole complex lab structure in the background. So you need super skilled technologists, you need proper reagents and quality control throughout. So it can be used certainly in a high resource setting, but it's a bit more challenging to use in some limited resource settings. And you certainly need a backup method if it's not going to work for whatever reason. So then there's a variety of automated systems, and the cool thing about the automated systems is that they all use slightly different technologies on the inside of the box, which I'm just going to go through quickly here. So everything from flow cytometry to like really high resolution optical microscopy. There's some interesting kind of biophysical detection mechanisms where like this microcantilever where the bacteria actually are stuck to it and it moves, and that physical signal is translated into growth. And then other methods use more of a metabolic activity approach where you're looking at the metabolic activity of the bacteria rather than the actual growth of individual bacteria. And so all of these systems are, again, decreasing that time to result for the AST by about a day. And then usually they're sort of modular, so you can run 1 sample at a time, but you might be able to get 4 machines. You could run 4 at once if you needed to. And these systems typically focus on gram-negative AST, not so much a broad spectrum of species. And they do all require a separate workflow for species identification, which can easily, generally speaking, be achieved in a large lab, which would have access to, for example, a MALDI machine that could do the species identification. So in our review article, we kind of tried to describe, similar to how you would describe pharmaceutical in its stages of development, we described these AST phases of validation. So everything from phase 1, which is just a proof of concept, sort of a beginning level study, to phase 3, which is the thing you would hand to the FDA or Health Canada to get your technology approved. And then phase 4, which is the post-marketing, like, is this a clinically effective tool or not? So I just wanted to have a look quickly at some of the phase 4 studies for the commercial technologies I've talked about, given that as a clinical person and a person who might make decisions in the lab about a technology, I'd want to see some evidence of whether they're useful or not, or whether they're accurate or not. And this is just showing in our review what we captured in terms of where we're at in terms of number of technologies in each phase. You can see the bars on the right there are the number of technologies, and the orange is actually FDA submissions and European market submission. So for these technologies here, basically the bottom line is we have understanding that they correspond well to reference methods, which is a good start. And then the time to result for these technologies is also pretty fast and comparable to what is stated by the manufacturer. And for these 2 technologies, we actually have more clinical information on actually reduced time to optimal treatment. We see for one of these technologies here on the right that maybe it's a bit of a mixed picture in terms of whether they found in their cohort it was helpful. So sometimes they had difficulty with the categorical agreement, so whether it corresponded to the reference method, and they didn't see clinical impact among every single patient. Okay, so for the last commercial technology I'm going to talk about, I will talk about Accelerate Pheno. So this is a technology that has the AST plus species identification, and the whole thing is completed in about 7 hours from the positive blood culture bottle. So they use gel electrophoresis to remove that blood matrix. Then they do identification using fluorescence in situ hybridization. And then they use some advanced optical microscopy to watch the individual bacteria grow while they're exposed to antibiotics. And the drawback here is they do use a very pre-specified panel of organisms and drugs, but it does have some good phase 4 evidence, so it's pretty reliable in terms of the results it gives, with potentially one exception with Pseudomonas there in the literature. And it clearly reduces time to result and time to appropriate therapy. There is even an RCT with this technology showing, again, their primary outcome being median time to first antibiotic change, and that being a significantly reduced time. Now, the study was not powered to look at patient outcomes, but that would be another important outcome to consider. And it's also important to note that there was a significant decrease in median time to antibiotic escalation, which was needed in about 25% of patients in both arms, 20%-ish. So this is just a quick picture showing basically time to result. Those top, so the top tests with the longer bars are some of the technologies we use quite routinely already in our lab. For example, my lab uses Vitec 2. So that's, it's going to take up to 72 hours to get your result there. And then some of the technologies I spoke about are the shorter bars. But still, you can see that we're not really getting under even 24 hours at this point, except for the direct from urine system, which is obviously has quite a specific clinical application. Okay, so now I'm going to talk briefly about a bunch of non-commercial technologies. Again, these are all ones that are going direct from specimen, so direct from urine to start with. So here, again, we see sort of a wide range of technologies used to get this rapid result. So often microfluidics are used, and then some sort of advanced either imaging or spectroscopy technique. So you can see that here for a couple of them. There's also some that use radioactive labeling, which I think would be a real challenge to implement in a clinical lab, so probably not as realistic of a solution. Others use flow cytometry, and others use electrochemical methods of detection of bacterial concentration or growth. So here's just in a figure form an example of one technology where there's a microfluidic device with an antimicrobial gradient integrated within it. The urine is just put directly in there, and then a dye ends up getting a color change with bacterial growth, and that's what's used to visualize with metabolic activity, and that's what's used to visualize the amount of bacteria for a given concentration. And then here's another example direct from urine. Again, though, they've thrown in that fluorescence in situ hybridization, so you know what the species is at the same time as you're running your AST. And again, it's a fairly straightforward method of determining the AST using just fluorescence to amplify that signal so it gets detected at a lower threshold or more sensitivity than you would with regular like optical density, which is what a lot of systems currently use. So direct from positive blood culture, again, the methods can be everything from radioactive labeling to more of an electrochemical detection method or advanced imaging. So here I'll show an example where we have antibodies holding the bacteria onto an electrode, and then reduction of a dye, which is also attached to the bacteria, the reduction happening as the bacteria replicate, and this generates actually an electrical signal that's detected. So this is one signal, a different way of detecting the bacterial growth signal. And here's another example where instead of using an electrochemical signal, this is actually just a physical property of essentially the solution with the bacteria as it either grows or doesn't grow. And what they do here is they measure not only the impedance, but also the something called differential pulsed voltammetry, so two different electrical signals, and the combination of the two signals is what increases sensitivity of detection. Okay, so for the final technology I'm going to talk about, it's very exciting because it's actually directly from blood. So this is, and this was published in June. So we've actually seen here that they took away the part where they incubate those blood culture bottles. So this is a really exciting potential technology, still obviously in very early stages, but this is where the time to result is actually just about 11 hours actually from specimen reception in the lab. So we've taken away that whole first 24 hours potentially. So to do this, what happens is the researchers took this special glycoprotein that can bind to any bacteria and added on a magnetic bead. And so the bacteria are actually bound up with this magnetic bead glycoprotein complex, and then they're taken out of the blood matrix. So that sorts out the matrix issue. And then they resuspend it in a culture medium, and they proceed to their AST, and then they also have the species identification component. So the species identification, it's similar to the other technologies we looked at. It uses species-specific probes to detect specific not only bacterial species, but also resistance genes, which is great. And then they also take that suspension and they do fairly straightforward AST in a microwell plate with actually semi-solid egg rows, and then they watch the bacteria grow in the different concentrations using an advanced microscopy technique. So again, they're looking at sort of single cell replication. And all of this had pretty good results in comparison to a reference method. You can see there's some disagreement, particularly with Klebsiella and ceftazidime here. So clearly, room for more validation of this technique, but it's very exciting and potentially could have a huge clinical impact if it's pursued further. So here is just a graphical representation of that time to result of all the non-commercial tests we had a look at in our review article. Many of them take still quite a long time, given that they're focusing on they haven't yet been able to implement their test with the clinical specimen. They're starting from pure colonies, but this would be sort of the phase 1 where they're trying to demonstrate the proof of concept. But at the bottom here, you see some very short bars showing very quick time to result, which is very exciting. So has progress been made in those two areas I talked about at the beginning? So number 1, reducing time to result. I think it's pretty clear that that has happened, and certainly some of these technologies are things that we could implement in a high-resource setting. I guess it's not entirely clear if there's a clinical impact for all of the technologies, because we don't have much of phase 4 evaluation, but hopefully that will be to come, and we'll try to be creative and leverage the data that already exists to try and understand whether there is an impact here. I think the workflow simplification is still a huge barrier. There's been some progress made, but this is going to be really challenging to implement. So what I would like to see basically is more phase 4 evaluation of these commercial technologies, and then from the non-commercial technology side, I'd like to see more a focus on understanding how we can streamline workflow and consider use in limited resource settings. So thank you so much for listening. And thank you very much to my co-author, Grace, and Dr. Yansuni and all of our other co-authors for their work. And I'd be happy to take any questions now.
Thank you, Keely, for an excellent presentation. Any questions?
Hi, Freeman from University of Toronto. I'm wondering, out of all of these technologies, a lot of them required multiple steps and maybe like transfers of samples from one container to another. I'm wondering what is the appetite for that kind of stuff in like the clinical setting? Like, how important is it that it's a one button go kind of deal compared to here's a chip, put in this chip, and then here put it under a microscope, another chip?
Yeah, so I think thanks for the question. I think it really depends on your context. So it's interesting because a lot of larger labs with more personnel are often more able to do the stuff that requires more hands-on time. But if you're in a limited resource setting or a setting, for example, in rural Canada, you probably want something that's much more hands-off. So I guess, and then for each lab, it becomes kind of, there needs to be an economic analysis and a use case made. So it's very context dependent, I think. And it's up to us in the bigger labs to kind of also sort of facilitate development of new things. And if we really like this new thing, then it can be something that's taken and made into more of more of a point of care in your patient type of test, I think. Yeah.
Sorry, follow-up question since no one else is here. What is, so a lot of these you have shown have limited panels, so maybe 5 bugs and 5 drugs.
Yeah.
How far does that get you? Does that cover like 90% of what you want, or is it actually a big deal that you're able to do like potentially all of them?
Yeah, I think it's going to become more of an issue because I think already in our lab we see it's hard to keep up with even new software coming out for existing machines and things like this. So yeah, I think that's a challenge. I think it would be nicer if we could make our own panels and things like this, because already, for example, if I see a multi-drug resistant Klebsiella, for example, we would do an e-test, like a gradient diffusion strip for these advanced antibiotics. So, and that's not going to be necessarily on the panel that's already provided by the manufacturer. So it is tricky. I think that's going to be a challenge for whether or not we would implement the test, if it's going to be useful to us or not. I think this is a question I would ask too. So yeah, it's important.
Okay, thank you.
Thanks.
We have time for one short question and one short answer.
Ori Solomon from McGill University. A lot of these assays have dyes for detecting the bacteria and growth and all that. So I'm wondering, for implementation in LMICs especially, is how stable are these dyes and these kind of assay kits? Because when we think cold chain, we think vaccines, but perhaps the question is, how stable are these? Would they be able to store them in clinics that are maybe not super accessible for extended periods of time?
Yeah, that's a great question. And I think the short answer is, like, maybe we don't know very well, but also probably they are requiring some degree of like temperature control. I think a good example would be malaria test kits. So often they get stored in all manner of conditions, and it can definitely cause problems with the result. So this is like an example of where I think we need to see that as a focus of research, like how stable are the materials, how can we store them? It sounds kind of silly to say that, but I think it's actually a super important thing to know.
Is this information not provided with the assay?
It is, but that doesn't, well, it'll say something like it needs to be stored between 4 and 20 degrees at such and such a humidity, but it doesn't say, well, if I accidentally, left it out in 30 degrees, like, can I still use it? So that's a valid question. Yeah.
So thank you very much for your talk, and great talk. And yeah.
Thank you.